Welcome to ASM Live from the American Society for Microbiology meeting in Boston, Massachusetts in 2014. 2014. I'm not that old. We're here today to have a conversation about surprising, important, and interesting aspects of microbiology. I'm Stanley Malloy. I'm your host today. And before we begin, I want to emphasize that this is really meant to be a conversation. So those of you who are here in our audience, if you have any questions, please come to the microphone and ask the questions. And if you're out in the internet world, please send your questions to #ASM2014. Our conversation today is going to be about changes in the oral microbiome that are associated with pancreatic cancer. And our two guests are Pedro Torres and Scott Kelley, both from San Diego State University. So, Pedro, why is this important?
Well, the reason this is important is because pancreatic cancer tends to be one of those really aggressive cancers, as what was thought of before, that had the ability to metastasize very quickly. But what we're quickly finding out is that this isn't the case. The case is that the early symptoms for pancreatic cancer tend to be subtle and often manifested in other diseases as well. Because of this, pancreatic cancer isn't detected until its advanced stages, once it's actually grown large enough to be able to interfere with other organs. So by the time you detect the cancer at those advanced stages, the survival rate is very low. In this case, pancreatic cancer being a 5-year survival rate on average of only 5%. So really being able to look at the oral microbiome, perhaps we could try to find a biomarker for this early detection to be able to increase the survival rate by detecting in its early stages.
But why oral microbiome? That's not an obvious thing to most people.
Yeah, it's weird at first, but with the initial, the way this started is the initial research saw that people who had periodontal disease tended to have higher instances of pancreatic cancer. So there was this correlation going on. So people want to keep on looking at it and see what was going on, and that's what we started to do. We started to go off of other people's research who started looking at this and seeing what's really going on, because we do know that periodontal disease is shifts in the microbial community in the mouth. And we're seeing this in people with pancreatic cancer, so could there be something that's going on in the mouth with people with pancreatic cancer?
So Scott, to do this kind of work really takes a team of people that involve cancer researchers as well as others. Could you say something about the team that was assembled to do this?
Yeah, sure. The idea really originally came from an undergraduate who was working in Kelley Duran's lab, who's also involved in the project at San Diego State. She'd taken my microbiology class and gotten really excited, and Kelley's too, excited about microbiology in general. And then she ended up working at the UCSD Moores Cancer Institute. And she had been reading some papers, and so she sort of put it all together because she's working with people with pancreatic cancer and knew the situation, read a paper that suggested the oral bacteria were different in people with pancreatic cancer. So she came to us and said, well, let's look at the community as a whole and see if there is a distinct difference. So it took her, talking to Kelley, bringing it with me because my lab does the culture-independent molecular tools to look at all the diversity in the mouth, not just what you can culture. And then working with the people at the UCSD Moores Center to get the actual samples, which is really, really challenging. So that's the kind of teamwork you need to get some really good clinical samples and get all the methods to be able to do this study. And then working with Pedro as well, of course, has been has done a fantastic job on this project.
Well, so what kind of methods were used to do this initial analysis to identify the correlation of microbes in the mouth and pancreatic cancer?
You want me to ask it?
Sure.
So the initial analysis from the first paper was culturing, where they actually isolated things in the mouth. And there's a lot of things they can isolate from the mouth. So they grew them up and saw what they thought was a difference in the number of bacteria of certain types they could culture between people with pancreatic cancer and without. But we know from for many decades now, actually, that there's many more microbes in the mouth that you can't culture than there are ones that are. And that's true of almost every environment that you look at. About less than 1% can be cultured in most instances. So what we're doing is using, all of them have DNA. All microbes have DNA. All viruses, a lot of viruses have DNA or genetic material. And that's really easy to pull out of, say, saliva or any environment. You can extract all the bacterial cells, blow the cells open, get all the DNA out. And then you can use the nucleotide bases of the DNA as sort of a barcode to type out the species that are actually there. So this is what we call the molecular methods. And so we're using a particular gene that all the bacteria have as a way to identify what types and proportions of species there are in someone's saliva. And that's, essentially what, in a nutshell, what we're really doing in the lab.
So Pedro, what have you found? What are the differences that look like they're important?
So what we have seen with our preliminary data right now is that we are seeing differences in proportions of Campylobacter species and Leptotrichia species that are significantly increased in people with pancreatic cancer when we compared it to other people that are healthy, people that have other sorts of cancers and diseases, and even people with pancreatic disease. And that's the really cool part, is the part that it's specific to pancreatic cancer. It's not just something wrong with the pancreas, like pancreatitis or pancreatic cyst, because we've compared it to those as well. But it's something that's specific about people with pancreatic cancer that's really causing these shifts in these bacterial species in particular.
So are you looking at mostly people with later stage pancreatic cancer? Have you been able to look at really early cases to get an idea of when the shift in the oral microbiota might happen?
So the patients that we get are just people that come into UCSD Morse Hospital, and they vary in the stage of the cancer. Some of these are in the very early stages. Some of these already their cancer has metastasized. But we are getting some that have pancreatitis with possible pancreatic cancer. So what we're able to do is right now we've collected their saliva, and we have all their information. We got a volunteer over there, and we're hoping the next 6 months to a year, she's going to be able to go back and look at the upgraded patient files to see if these people that at one point only had pancreatitis did turn into pancreatic cancer. And then we'll look at their community profile and see, was there this change early on that we could have had detected with this method?
So before we go on, I'd like to remind people in the audience to please come ask questions at the microphone. And if you're out in the internet world, send your questions to hashtag ASM 2014. So if you guess from your results so far, and I realize you have a lot left to do, how early do you think that this change might happen?
So we have to be careful here. This is based on very early preliminary results. It is following up some other studies. And you get a limited number of patients in these studies because they're very difficult. So we're getting around 14 or 15 that actually have been ultimately diagnosed with pancreatic cancer. So we want to be careful to say we don't have a diagnostic test yet. But it's something we're working towards, and we still are collecting data. Anyway, what was your question again?
How early you think the detection?
Yeah, to answer your question, I don't think we know yet. We're hoping, of course, that we can detect it really quite early. I mean, I feel like the mouth, looking at all the things like diabetes and obesity and cancers and chemotherapy and all these other things, the mouth is almost a signal of greater health in the body. It's connected to the body. I mean, we have dentistry and we have medicine, but the body it's not split like that. And we think that other issues can be reflected in the chemistry and the microbiology of the mouth. So we're hoping that we'll see more subtle shifts. And maybe you can go to the dentist's office, get a saliva test, and it'll tell you something. That's the real long-term hope about your overall health picture and your future prognosis.
That's great. We have a question from the audience.
Hi, Alex Kostick, Broad Institute, Harvard Medical School. Yeah, these results sound very interesting, and especially the result of leptotrichia being increased. Leptotrichia falls in the Fusobacteriaceae, which is a small family, which the other major genera is Fusobacterium, and Fusobacterium, a number of labs have shown recently, is associated with colorectal cancer. So I guess I have two questions. One is there whole genome sequencing to confirm that it is in fact Leptotrichia and not Fusobacterium, because they are closely related, especially in the 16S? And second, what do you think the relationship might be here between colorectal cancer and pancreatic cancer?
All right, so for the first question, what we're doing to really be able to identify these species is we're using CHIME analysis. So once we get all the sequences, what we're doing is we're trying to group them in order of having 97%, at least 97% similarity, if not more. When we group them in this way and we blast it against, we put them against the machine, the blast machine, it's showing us that this is a leptotrachea species. There is always a possibility, it's not perfect, there's always a possibility that it could be that maybe another similar bacteria to leptotrachea got stuck in there with that group, but we're fairly positive this is the leptotrachea species that we talk about. I have heard that it's been associated with other things like colorectal cancer, and what is its association with it? It could be, I mean, it's really difficult to say, because the thing that's really weird about the pancreas is that the pancreas has many functions, both has endocrines and exocrines. And part of the exocrines do go out and help with the facilitation of being able to break down food and everything in the colon and the intestines and everything like that. So it could very well be that what we are seeing from the pancreas has something to do with the way it's influencing the colon, the small intestines like that, and that's why we're starting to see this connection. That's kind of what we're thinking right now.
So that's this connection between potentially the colon and the two types of cancer, colorectal cancer and pancreatic cancer. But let's go back to this basic question. Why would very early stage changes associated with the pancreas, why would they affect the oral microbiota in this kind of way? Do you have some ideas?
The first thing, someone brought this up when we were giving a talk, and one of the things that the pancreas does is make amylase. And now it's serum amylase, not the stuff in the mouth, but that breaks down sugar. And you can imagine if you did have, and I'm not sure the exact relationship, but if you did have a dysregulation of amylase in the mouth, that would change the carbohydrates and sugars in the mouth, and that certainly would have a big impact on the microbiome. So that could be, that could help explain why you would see an early stage change in the shift in the community and why that might be useful. As is possible that leptotrachea has something directly to do with the cancer, but I think it's more likely at this point that it's a systemic effect, and that you could see this if you, how early depends on how dramatic and how much damage there is to the pancreas, I imagine, to see how much you're going to see a shift in the bacterial community. So
I think that this is a difference, as I understand it, between what people are thinking about the colorectal cancer, where they believe that the fusobacteria may actually be involved in the initiation of colorectal cancer in some cases, whereas you think it may be an effect of pancreatic cancer.
I think that, with the fusobacterium, you've got a direct line to the colon in some ways. It goes right down. I don't know quite what the direct line, it's not quite a direct line. I'm not I don't know anatomy well, I'm a microbiologist, but I think it more of a symptom. But it's possible, it could go either way. I mean, we're doing a causal relation. We can't say causation, this is a correlation, and you can't infer the two. But either way, it's, it would be a great marker, and further research will need to be done.
We have another question from the audience.
Hi, Andy Cutting, SASU, UCSD. I'm curious as to the fact as you guys looked at whether human genetic variants actually attribute to the shift in the microbial communities within the mouth, which is the case of Crohn's disease. They showed that in the small intestine, they know that polymorphisms in autophagy-related genes actually contribute to the pathogenesis of this disorder, and do you think that this could actually be applied towards this type of research, connecting human genetic variants and microbial shifts in the mouth?
So,
yeah, go ahead. Oh, I can try to answer. So you're saying, well, we don't have, unfortunately, any data on any polymorphisms in the patients that we're looking at now. So we can't do that correlation. I think you need a fairly large sample size to really see that with this data, but I wouldn't rule it out that there are some genetic factors involved with both. With the preliminary results we're looking at right now, it seems like the shift is strong enough, if it holds up, that it will overwhelm any of the genetic factors of disease. This is what we see. Yes, we have a good chance that you're going to come down with pancreatic cancer, or so I'm not sure, but right now I don't think we have the data, but I wouldn't rule it out for sure.
Do you know of any other examples where changes in the oral microbiota are a reporter for another type of disease?
Yeah, there have been early studies suggesting that changes in the oral microbiota could be early indications of oral cancer, which would make sense because it's happening right there in the vicinity. There's also been other studies that suggest that changes in the oral microbiota might be related to lung cancer and things of that sort. A lot of this is early. There's nothing that's, okay, this is what's really going on. but there have been other people looking at different parts of the body and seeing if they could find a biomarker for that as well.
It's interesting from the previous question to think about the human genetics in addition to the microbiome. And one of the things about a number of these diseases is that there are strong correlations of the cancer in certain groups, certain ethnic groups or certain socioeconomic groups. And I wonder if there is a way to begin dissecting that type of information in the samples that you have for pancreatic cancer.
Yeah, actually, what's really great that we are getting these patient files is that we do include things like ethnicity, gender, age, and other history like that, smoking. And when we took those variants into consideration when doing the analysis, we saw that ethnicity didn't really correlate with them having certain microbial community, like the way we saw it in people with pancreatic cancer. Now, there is higher instances of African Americans developing pancreatic cancer, but when we threw that in, but our sample size of African Americans wasn't large enough because we're sampling from San Diego. Not that many people are going to the UCSD Morse Cancer Center, but we do have a lot of Hispanics, a lot of things like that, and we didn't see any sort of correlation that said, okay, these people, we're seeing this microbial shift in people with pancreatic cancer, but we're also seeing a similar microbial shift in people with this type of ethnicity or that type of ethnicity.
So if you think about this as being a test that could be used clinically, let's just say the dentist's office. We don't normally think of the dentist office as being able to do DNA sequencing. Maybe that will happen soon. If at least some companies that make DNA sequencing equipment would love the idea of selling them to the dentist's office. But you view a chip type of test? Or?
No, well, I mean, one of the things, if it's really simple in this case, it could be, other cases could be much more complicated with diagnosis, but if we're seeing a shift in two or three different particular bacterial species, it's fairly straightforward to have a little, say, PCR test that targets, which we're kind of doing right now. It's like, we're going to target just Leptotrichia. We know this little PCR primer hits that, and you could do a simple, detection ratio between, say, Leptotrichia and the total kinds of bacteria that are there, and see if it's really shifted. So I'm hoping it would be something as simple as looking at a relative ratio with a particular marker that you could easily put in, instead of having to necessarily sequence all of the DNA of all the bacteria, which actually could be really possible soon. But it's also very difficult for interpret. But a quick interpretation of ratios of bacteria, I don't think that test would be all that challenging.
But this ties into something that dentists would be particularly interested in, because early on when I said, why did you begin looking at the mouth, you brought up the concept of periodontal disease, right?
Right.
And so dentists deal with a lot of periodontal disease. And so the question is, are these changes associated with periodontal disease, or are they associated with pancreatic cancer? Can you distinguish those two things?
Well, this is okay, so we're looking at saliva, we're not getting out of gum pockets, and the correlation between what's in the saliva and how much periodontal disease you have, I'm not sure that's established, especially with our methods. But from what I'm seeing, it's such a dramatic shift that it is, even if you had, all the other people that we're seeing, it's, all the different cancers and healthy, it's just variable ratios of bacterial that are normally in the mouth. And I think it's well beyond what you'd see with just periodontal disease. Of course, in the future, they would love to take some saliva and say, how bad is your periodontal disease? Which is something we're kind of working on in our lab in a different study. But I do think, to be really effective, it's got to be a completely different pattern of bacteria.
I think that's very interesting, because periodontal disease has also been associated as a causation factor of certain types of diseases, like heart disease.
Right.
And
working on that too.
But if there was a way of distinguishing the periodontal communities associated with each disease, now instead of one reporter, you have many reporters.
Right. One caveat on that, we're also finding that individuals are very different in even their response to periodontal disease. So it's highly important for most things. In this case, maybe not, because it's such a dramatic shift. But in many cases, you have to take the individual's variation into account and maybe even follow them over the time, because two people have very different responses and to say treatment of periodontal disease because of their underlying probably immune system. So it's going to be an interesting, it's complex field, periodontal research. But this one might, hopefully, is a simple one where you think, look, it's this shift. You are going to come down with pancreatic cancer. Let's treat you now. That would be really, really ideal if we can come up with that.
Is there an animal model that you can use to also look at these types of issues?
We haven't begun looking at any animal models right now, but there's been other people who've looked at mouse models and things of that sort to look at other effects that pancreatic cancer might have. So there is that also possibility that we could look at the mouse model and see if we're seeing any sort of shift, but at the same time, we don't really know what their oral community is like as well. So we don't know if we're going to see the same exact pattern.
Yeah. I mean, this is one of those cases where you really have a very valuable human population that you can look at because of the seriousness of pancreatic cancer.
That's a tough one.
And so what next? What are you going to do to extend these studies to go to the next level? Is it just more or what?
It's a combination of more and using other techniques as well. So more as in we want to try to get more patients that come in to really see if what we're seeing really holds up. Right now it's a relatively smaller sample size. We do want to get more people in here to add to this experiment. But also, as mentioned earlier, we were thinking that maybe salivary amylase and things like that might have something to do with these shifts. So we also want to look at salivary amylase levels between different patients and see is this really going on? Or are we seeing that people with pancreatic cancer do have a shift in their salivary amylase? So we could say, okay, this is a correlation. This is something going on right here. We're seeing this dramatic decrease. We're seeing this increase in this bacterial species. It's a salivary amylase and doing things like that. And going along with what Scott said before and really starting to look at more molecular techniques now, as in qPCRs and things like that, to see if these shifts that we're seeing are really there. To look at these shifts and try to go towards this biomarker phase.
That's great.
Yeah.
We have another question from our audience.
Hello, my name is Brittany Demmett from the University of Colorado at Boulder. You may have mentioned this before and I just missed it, but can you speak a little bit more about the demographics of the people you tested as regarding age, since that has an effect on the oral microbiome, as well as how many of the people tested had periodontal disease and all of those with the different cancers, were they all recruited? And just more details on that. Thank you.
Yeah. So the average age is around 60 years old. There's going to be the occasional person who's really young going there, but because this is a cancer center, people with cancer tend to have it at the later stages, especially pancreatic cancer. So a lot of these people are around that age. I'd say it's around 60 years old when we average it out. The second part of do we have their periodontal data, periodontal disease, things like that, we don't have that. That wasn't part of the questionnaire when we first initially started asking, and that's not something that is shown in their patient files at the doctor's office, at least MD.
But so to expand these studies, one of the things that's pretty nice is you just need saliva.
Yes.
Right? You don't have to do surgery on somebody to get your samples. So potentially you could really get samples from different parts of the country and have them sent in to be part of the collective analysis here. But the real challenge, as I understand it, and I think ties to this question, is the metadata that's associated with this. Does the person have periodontal disease? Are there other factors that could be very important in the interpretation? Is that correct?
Yeah, I mean, it's, like you said, trying to bring samples in from other parts, that'd be difficult in itself, trying to get that going. But there is a lot of other factors in there, in that we don't really know if they have periodontal disease or not, and I forgot what I was saying this question, but we don't really know if they have periodontal disease. That's pretty much what it comes down to. It'd be very simple, though, if from now on, when we are collecting these patient samples, we could just ask a simple question. Right. Do you have it? Have you been diagnosed with it? How long away are you getting treatment for it? That'd probably be a little bit easier way than having to go through different doctors and trying to figure out exactly their entire history and things like that.
Great. Another question from the audience.
Hi, I'm Jonathan Guy from Youngstown State University. I was wondering about the sampling of the saliva and also what methods you used and which ones you'd like to include in the future.
So part of you could answer the saliva question.
So just how we're collecting the saliva. Just patients that are going into UCSD Morris Cancer Center, they go in, we had a volunteer there, it was Erin Fletcher, and she would go and just ask them if they would like to participate in the study. They're really only going there for things like endoscopies, colonoscopies, And if they say they want to volunteer, we just hand them a tube, they sign the voluntary forms, and they just spit in a tube. And it's really that easy. That's what's great about just using the saliva.
And you're interested in the molecular methods past that? Sort of the, yeah, so once we have the saliva, there are some really standard, it's really become very standardized, the DNA extraction methods for a whole community of bacteria samples. And so we essentially use a really standard kit that everyone uses. So if there is a bias in it, everyone has the same bias. And right now we're using the mobile soil kit. It sounds really strange, but it's really good at mechanically shearing all sorts of different bacterial cell types, and archaea actually, if you want to go there. And then once you get the DNA purified, well, DNA is sort of DNA. I mean, it's negatively charged, it's easy to work with. Then we use, for the gene, when we target a single gene they all have, we use these universal bacterial primers that essentially bind to all the 16 of all the bacteria in a sample. And that, there are some biases inherent in that, but once you get that amplified, we use bar-coded primers, which allow us to do many, many people at once and pull them all into one sequencing reaction, and then, sequence them and reinterpret the data about what bacteria are there and how many. Yeah. Sorry.
And, which universal primers do you know off the top of
Yeah,
your head?
515 forward and 805 reverse, which is the sort of standard set for the Earth microbiome projects and the other ones, which is nice because to be able to compare data sets, people have all sorts of different primers and they have different, but this one's been pretty good for the oral microbiome community. It captures a lot of the members of that community, probably not all, but it certainly captures well the ones that we think are really different.
And the volume of the saliva collected?
So
that can vary.
Yeah, it really depends on the person that's coming in. Sometimes it's really nothing that we could even work with. Sometimes it's a lot, but all we need to be able to isolate the DNA that we need in order to get sequencing is only 55 microliters. But even for some people, that's difficult sometimes.
And you mentioned sequencing and PCR. Are there any other methods that you use to look at
We're going to do whole genome, metagenomic, which is to shear up the DNA into little bits. Of course, you need enough DNA, and I think someone is asking about other methods we're going to use. But we're going to use that as a background because that doesn't have the amplification biases, and we're going to see if we have the same sample. We're doing this first round now, but we're going to go back to most of those samples again and sequence all the DNA from all the bacteria through the metagenomic sequence. Should be a little less biased. But of course, you have to have enough saliva. Some of these people, actually, saliva is difficult. They're doing chemo. Their salivary glands are producing a lot of saliva. It's tough. So we're working with what we have, but it's going really well so far.
We have a final question from the audience.
Hi, Ilya Chorny from Illumina. So have you, to expand on your comment, have you thought about doing the metatranscriptomics? Because there's evidence to suggest that the genes might correlate better than the community structure. You might have different community structures, but similar genetics or gene expression structures.
Yeah, I think there's a lot of things like periodontal disease and other sort of active bacterial communities where the bacteria are heavily involved in the process that's going on. I'm not so sure that this is the case. So when you really need to know what genes are turned on at this thing, I think in this case, this is my guess, it's more of the types of bacteria that are present, and it's sort of a signal. This is what happened to your community, and to me, it's less about what that community is doing than what it is. So I think that would be a really good thing to do, and we could do that in the future, but trying to figure out who's there in this particular case may be hopefully all we need. Because if we have to go to the metatranscriptomics, it's going to be tough to get a diagnostic. But we might end up there.
Well, so this is a great example of where our development of molecular techniques has opened the doors to ask questions that we wouldn't have asked before. And also a case where a simple technique like this may save a lot of people's lives by providing early diagnosis. It also emphasizes how much more has to be done and how hard it is to get good sample sizes to know definitively about these types of things. Thank you very much for joining us. Scott Kelley, Pedro Torres, both from San Diego State University. I'm Stanley Malloy. Thank you in the audience for joining us today, and all of you out on the internet. Join us again for the next version of ASM Live.
Thank you.
Once you're in the BSL-4 space, the only way you can get out is to go through a chemical shock.
It's an unusual room, never seen anything like this. Anybody who has access to this facility first has to go through an R scan. So
The HEPA filters filter the air coming out of the facility, and that will remove bacteria, viruses, anything that might constitute any kind of risk, right?
Remember, this building is basically a second building inside the main building.